In this video, I'm going to be going over how to open some basic scripts in R, the difference between a normal .r file and a .rmd file, or an R Markdown file, how to generate HTML or PDF outputs for your files, and kind of the overall usability of this environment and how you use each of these different panels. So let's start by opening up our file. I'm going to start by opening up one of these .rmd files, or R Markdown files. And this is basically going to be a script of instructions, and this is going to contain a header that is basically only for the HTML portion to be able to generate a document, along with the first line in each chunk as well, and the very last line of each chunk are these three little tick marks. So for each section or chunk, you have a line here telling the HTML document whether to hide it or show it, whether to keep figures, whether to print things out, and this to end the header. So that creates a chunk. So let's compare this to a normal R file. Now this is the same R file, but it's all one giant chunk. And when I say one giant chunk, I mean you can run the entire thing. If you hit run, it will go through every single line. And there are kind of sections in it that you can make by using these hashes. These hashes allow you to make comments. If you take them away, the program thinks it's a command that needs to run, and it will give you an error. But if you add this hash, you can put comments in, you can break it up into sections, you can kind of start to make your own structure. But for me personally, I find this a lot harder to work with. So I prefer to do .r Markdown files. Although you could copy any of this and paste it into a normal R file if you like it for simplicity. But let me go ahead and tell you about the HTML version of this. So for instance, after I make this entire analysis and I generate graphs and data, it outputs an HTML file that looks like this. So the very beginning shows the title, which is here. It shows some text about who made the script and where to look for resources. And then this very first chunk shows all the libraries that you will need to run the program. And a lot of these algorithms rely on individually written libraries, and so a lot of them are from Bioinformatics here. So if you don't have this installed, you can unhashtag this and make sure you install Bioinformatics, and then some of the programs like DE-Seq2 is important, Annotation Hub is important, etc. So if you want to create one of these HTML files, you're going to need to use an R Markdown file. And these are great because they have table of contents that are navigatable, so you can go directly to a section, particularly if you're talking with your boss or collaborators and they want to see, for instance, the heat maps between different groups. You can go ahead and show that. If you want to look at the volcano plots, you can look at that. So here are the volcano plots. And the great thing about this is it doesn't just display the graphs, it also shows the code that was created to write that graph. So this then becomes a valuable learning resource for you and your lab to know and to exchange and to collaborate and write new code, do new contrasts for your groups, and to be able to work within this code. For beginners, it's a great opportunity to learn what happens behind the scenes to create these graphs and how to actually go about doing the analyses. We use the pipeline from the Harvard Chan Bioinformatics Core website, and there's a link to that in the header. They have done an excellent job at outlining resources and showing why certain quality control steps need to be taken. The HBC posts all their content online, so you can take your own course at your own pace and learn more about RNA-seq analysis. The first thing you're going to want to do is check your raw data to make sure there are no clear outliers, that the distribution of your RNA data matches what's expected, and you're not violating any basic assumptions. For instance, most RNA-seq data follows a negative binomial distribution, and that assumes that the mean counts do not equal the variance of the counts. Using an R Markdown script, just ignore the header of each file. So all this means is training your eye to ignore things that aren't essential. So that would be ignoring this here. You only have to worry about installing Bioinformatics when you're first starting. otherwise you should have it. And then we have a bunch of libraries here that need to be loaded. You're going to also need to set your working directory because R needs to know where to look for your files. So in this case, I have a folder on the desktop called RStudio, CSR, and osteocalcin. You can see if you navigate here, RStudio, CSR, osteocalcin. So now I'm in the main folder where my data is. I have a results folder where I will output CSV files as well as plots. And if you want to set this as your working directory, you can click this little More button, go to Set as Working Directory, and now you see it appear on this console. So you can copy and paste this into this line here, and of course this is going to differ for your computer. So that works. Another thing I'd like to mention is that it's really important to have reproducible methods in bioinformatics, because everyone's computer is different and packages are constantly changing. In order to be able to reproduce your digital methods, you're going to want to record this information. So for instance, session info is this command to record everything, and I should run the library first. So first I'm going to go ahead and run this first chunk here by pressing that green arrow. This is going to load all my libraries which I have previously installed and I know to work. And basically once you get that session info, you can write those lines and have it recorded, and it'll show all the packages that you have installed and in what versions everything is in. So now let's do this. Methods. Okay, so now this is going to show my environment. Also, when you're troubleshooting, if you want to upload to any sort of forums like BioStars or Seek Answers, a lot of times they ask for your session info. So that's how you would get it. It shows all the packages I have installed, what version they are. So this is a very important information to keep lab methods. Like any other thing in science, bioinformatics is an experimental region that needs to be recorded as well. Okay, so that's just the very beginning of how you're going to load scripts and kind of understand things.